Okay. While we're setting up, I'd like to thank you for inviting me and thank the organizers. And I want to say that you're in for a shift in gears. Thank you. What I'm going to be talking about today is not neurodevelopment, some links to neurodevelopment, some links to neurodegeneration, but primarily focusing on learning dependent plasticity in the adult human brain, so models of GABAergic and dopaminergic systems of the human brain and how they support learning and brain plasticity rather than cellular and molecular mechanisms. So please hang in there, and I hope you'll find something interesting. For the past 15 really years in our lab, we try to understand how the brain solves the problem of extracting structure from our environments with which we interact in our everyday lives and making sense of them. And that's not something that you do only if you're kind of daydreaming and looking for planets and galaxies in a nice, clear night sky. It's something that you do on your everyday life when you're trying to learn a new language or navigate a new city or learn about music. It's something really that we do in our everyday lives. And I'm sure you won't be surprised if I say that you'll do better if you practice. That really practice and experience with the environment makes us better in all sorts of quite demanding tasks. For example, when you're looking for targets and making decisions in very cluttered and noisy environments. Or, for example, when you're trying to learn a new language. We know from seminal studies in developmental psychology that very young infants very quickly can pick out from streams of nonsense speech patterns that are frequent versus infrequent. So how does the brain do this? When we usually try to investigate brain plasticity in the brain, we use either neurodevelopment models or interventional models. So obviously we know that there are amazing changes that are happening with development in the brain, and we can study how the brain changes in terms of its systems during development. And we do the same when we apply interventions, for example, rewiring centers in the brain or peripherally rewiring and affecting the brain organization. But when we ask questions about how everyday experience changes our brains, we have rather subtle changes. So how can we go into the brain and understand how its organization changes through experience with the environment? To do this in our lab, we recruit many different methodologies and combine them. So we start with human behavior in the context of complex tasks, and we take advantage of brain imaging technology, different types of multimodal imaging, together with computational approaches that allow us to extract rich information from biological data. So the questions that we focus on, I'd like to give you a feel about today, is we look at brain systems that support our abilities to learn about spatial structure in the environment so that we can interpret the environment and deal with it, interact with it. But also we look at how we learn temporal structures, for example, as I said in the context of language, to be able to predict what's going to happen next, what is the next thing that is coming up. So through several studies using functional brain imaging in our lab in the past, we have shown that, in fact, humans have an amazing capacity in a very complex environment to identify what are the critical features to which they need to pay attention to solve complex tasks. For example, in the context of categorization, of visual categorization tasks, if you're trying to figure out what does this look like, and it's a completely unfamiliar and novel type of object that you're experiencing, your brain actually can identify key features that differentiate this object from other objects you might have encountered or find similarities. And in fact, what we see is that this ability to be able to do this quite fast and successfully relies on specific circuits in the brain that are involved both in sensory processing and in perceptual decision-making. And these circuits actually reorganize and rewire in terms of their connectivity and in terms of their processing through learning and experience. But today, really, what I want to focus is a new technique that allows us, in fact, to look at GABAergic inhibitory mechanisms in the brain. We heard about interneurons. We cannot access interneurons at the level of the human brain with brain imaging techniques. But thanks to developments in MR spectroscopy and thanks to talented physicists both in Cambridge and Oxford and colleagues at the University of Oxford on the 7 Tesla scanner, we have started this work in using MR spectroscopy to measure GABA in the live human brain. So what do we know about GABA and how does it relate to learning plasticity? We know that GABA actually decreases in GABA relate to plasticity. And in fact, we know that imbalances in GABAergic mechanisms and excitation, so GABAergic inhibition and excitation, actually relate to neurodevelopmental disorders. So here what we ask in these studies is really how GABAergic mechanisms support our ability to make sense of the environment, to make really difficult judgments in cluttered scenes. And to kind of simulate these environments, we create type of stimuli like what you see here that are nicely controllable. So we ask our participants while they're in the scanner to tell us whether this pattern looks like a radial versus concentric. Obviously, this is easy, but it's much harder if I put noise. So if you're in a busy environment, can you make this judgment? And it's also very hard if I morph between these patterns, I create kind of stimuli that are very similar to each other. So we put individuals in situations where they have to make really difficult decisions about sensory information, and we see how really we measure their GABA during training in these type of tasks. And what we see is a classic signature of plasticity where we see that GABA decreases in sensory areas of the brain as people become better in one of the two tasks, in the task where they are trying to identify patterns in cluttered scenes, for example, finding a friend in the crowd. However, what we see is the opposite pattern, an increase in the GABA with training when individuals are making difficult judgments about discriminating patterns that belong into different categories. Now, these mechanisms link actually back to animal models of plasticity. I don't have time to discuss this more, but I can explain more later. Interestingly, what we see is great variability in GABA concentrations in humans, but also great variability in their performance in terms of how much they benefit from the training in these tasks. And we see interesting correlational patterns. What you see is when you detect a friend in the crowd, more decrease in the GABA makes you better in the ability to learn this task, while if you're trying to discriminate very fine differences, to tell apart, for example, your friend from a twin, then increases in the GABA correlate with behavioral improvement. How does that relate to what we use to measure in terms of functional activity in the human brain, which is accumulated processing of both excitatory and inhibitory processing? So we see exactly the same dissociation when you are looking for a friend in a crowd. Decreased GABA relates to higher bold signals. When you're identifying very similar patterns and discriminating, increased GABA relates to higher bold. Now, what about connectivity? I said that actually what we see is that the brain actually through training and experience in the environment changes its connectivity. What we see here is that these GABAergic mechanisms that support our ability to learn actually relate to changes in connectivity. Local connectivity relates to decreased GABA and supports our ability to learn in clutter, while connectivity between sensory areas, in this case visual cortex, and decision-related areas, in this case parietal cortex, actually increases as GABA also increases to support our ability to make very fine discriminations and perform difficult judgments. So just to summarize, if you're if you're looking for discriminating between colleges in Oxford versus Cambridge, you actually need your GABAergic inhibition to help you become better in this task. We actually see that GABAergic learning-dependent plasticity changes in a very kind of task-dependent manner, depending on what really are your interactions with the environment to support our ability for complex cognitive tasks. Now, moving on to another space in terms of how do we learn temporal structure in our environments. Our environments span both space and time. We're looking here at different types of tasks that allow us, for example, to extract patterns from sequences. In our everyday experience, the environment is dynamic. We always deal with events. One comes after the other. If you are trying to interpret the music you're listening to or a language, or the first time you're learning a language, that's exactly what you're experiencing. You're experiencing a sequence of events. In the beginning, it doesn't make any sense. Through training and learning, you acquire this capacity to parse speech, to identify events, and also to make predictions about what's going to come up next, which is obviously very important when we deal in complex environments. So how do we do this? We devise paradigms that are very different from what I showed you before. We set a new task for our participants, a new problem. In this case, we show them completely unfamiliar stimuli, like these symbols that you see here. The symbols appear one after the other. At some point, the sequence stops, and our participants have to tell us which one of the symbols they expect to come up next. Now, what we do is we always play a trick. Psychologists like play tricks. We change the order. We determine the order with which these symbols appear, these events appears. And in fact, we can use Markov models to make these orders as easy and as more complex as we like. We start in an adaptive training manner, so we start with very simple orders. For example, one of the symbols is more frequent than all the others. Then we have pairs that are more frequent than the others. Then we have triplets that are more frequent than the others. So we deal with, we increase the complexity, and we try to simulate situations in the environment where the structure of the environment is hierarchically more and more complex. And then we ask, well, can people actually learn? And actually, they learn without a teacher? In this case, we do not give them any feedback on a per trial basis. We just let them loose in this complex environment and see what they learn. And in fact, what people do is sit in front of the computers, look at the sequences. At some point, sequences stops. They have to figure out, to be able to predict what's going to come up next, they have to figure out what is the structure in these patterns and what are the exact combinations of symbols that appear. So how do they do this? They do actually quite well. But the interesting thing again here, there is individual variability. You see, some individuals are faster than others in extracting this temporal structure. Similar as I was talking earlier about spatial structure, we see variability. Now, in the past, we tend to ignore this variability. We would report it, but we would average, and we talk about the average brain and the average cognitive functions and plasticity. Now we do have methods through computation, through techniques that are developed in terms of neuroscience methodologies that are developed that allow us to look into the human brain, and that's what I'd like to show you. That's exactly what we've done. We went after this individual variability to understand the capacity of each individual to learn, because this is very important when we are looking at interventions, rehabilitation programs, and treatments. So what you see here is that in fact, individuals differ. Some people are faster in identifying structures, some people are slower. And what I'll just show you is that this relates really into the decision strategies that the individuals have. It relates on how they approach the problem. What is the problem-solving strategy that they employ? If you look in animal and human choice behavior, we are categorized as either matchers or maximizers. This means we either try to learn about the environment and learn about its statistics and adjust our behavior to these statistics, or we maximize. We try to make the best out of the reward. Whatever is more rewarded or whatever is more frequent, we will respond to that. So these two behaviors, we looked at them and we plotted participants' performance against these models. And what you see here is that the blue people that tend to be the slower ones are the people that stick with matching. They try to memorize everything about the environment, learn the full structure of the environment. However, the people that realize that this is a quite difficult problem start deviating from this matching strategy towards a maximizing strategy that allows them to respond based on what's more frequent or more rewarded. And what you see is that they learn faster. And there's a strong correlation between the strategy the participants adopt and their capacity to learn. And so we went to look in their brains because at the same time that they were doing these tasks, we had functional brain imaging data, we had connectivity data, both structural and functional. And when we looked at the brains of these individuals, we realized that actually they use completely different circuits based on the strategy they adopt for solving these problems. These are the same individuals. We did not know how they approached their strategy. We modeled their behavioral data, and then we used the parameters from these models to analyze the brain data. And what we see is that when our individuals are using a matching strategy, we recruit the hippocampal visual circuit, while when individuals are using a maximizing strategy, we use they use frontostriatal circuits. And we see this in functional brain imaging studies. We also see it in connectivity studies. So if we look at structural connectivity with DTI and we look at changes that are happening in structural connectivity, we see that a matching strategy, again, relates to changes in visual cortical striatal circuits, including the hippocampus. We see that a maximizing strategy involves frontostriatal circuits. The same thing we see through functional connectivity. We map the striatum and we look for connections of the striatum to cortex, and we look at changes in these connections and we extract exactly the same circuits, visual cortical striatal circuits for matching, frontal striatal circuits for maximization. So I even started believing this pattern. And then we looked at graph theory analysis in terms of identifying more closely the different nodes of the networks and their connections, and we see exactly from the circuits that I told you that we can predict the behavior and we can predict the strategy of individuals. Now, why is this important? What we seem to be identifying is that the brain actually can have alternate routes in solving the same problem, and seems to evoke them and use them in a very different way in the same individuals. One route goes through hippocampal and visual circuits, in this case sensory circuits, for more memorizing. Another route goes through frontal striatal circuits. Now, does this mean that if we have disease, if we have neurodegeneration, and one of these routes is damaged, maybe the other route could be exploited, and therefore behavior could be remediated? To test this, we looked at early stages of Alzheimer's disease. We looked at mild cognitive impairment. We selected patients that had hippocampal dysfunction. And these patients, we see that they improve in this task the same way that our controls improve. And what we see interesting from their brain imaging analysis is that they use frontal striatal circuits rather than hippocampal circuits, which we see more in control, healthy control individuals, suggesting potentially that when one route in the brain is dysfunctioning, the other route can potentially take over with obviously great potential for rehabilitation. So to summarize, I hope I've given you a flavor that it's not only neurodevelopment that relates to plasticity. We have extreme plasticity in the adult brain across the lifespan. And we see very different mechanisms that can support this plasticity, both GABAergic and potentially dopaminergic reinforcement learning circuits that can support this capacity for plasticity in the adult human brain. And very important, the focus is now on individuals and how they approach their learning and what this means for the brain circuits that are potentially involved. I haven't touched on any other predictors that potentially are involved in capacity for brain plasticity and learning in individuals. There are many of these factors that you've heard about in these days, from molecular and cellular factors all the way to environment. And by working now in large neurodevelopmental cohort studies, we have the capacity to really bring together multimodal data from brain imaging, from behavior, from genetics, from all sorts of omics that allow us through advanced computational approaches, for example, in the field of AI and machine learning, to start mining this data, understanding predictors, but also mechanisms of health and disease. So I'd like to close by thanking everybody in my lab that has made this work feasible and great fun, our collaborators, the people that give us the money, and you for persevering. Thank you.
Hi, thanks, Zoe. Those two circuits that you pointed out, the striatal, prefrontal, and the hippocampal one, I'm not sure because it flashed by a bit quickly, but it looked to me like the maximizers, which would be the optimal learning strategy, maps onto the striatal circuit, and the matches maps onto the hippocampal, but the MCI patients seem to be using the better strategy. Is that right?
Yeah, so I did go a bit fast because my slides were off going 5 minutes, 1 minute, and I kind of giving the general message. Yeah, so it's really interesting. I kind of avoided to talk about optimal strategies because actually both strategies could be optimal in different environments and in different situations. And we see a very interesting pattern in terms of timing. So initially what we see is that the people, if you want, that perform the best are people that initially everybody seems by default to start with matching. Maybe it's because we do have hippocampal and frontal structures that support this. Maybe it's because our educational system makes clear that we memorize our timetables from very early on and we stick with this. Then what we see is that people switch to maximizing to solve the problem. And then if we keep training them, we see that even the maximizers, they will switch to matching to be able to actually learn the whole statistics of the environment. So there's more of a kind of a dynamic. And the same way the circuits have a dynamic into them. So I didn't go into detail, but the visual corticostriatal loop actually involves caudate, hippocampus, and visual cortex. In this case, because our stimuli are visual. So it has part of the striatum, and it has the caudate, which we know that together with the hippocampus is involved very much in pattern matching. Now what we see is that potentially the role of the frontal cortex here, the different structures that we see, play the role of switching the strategy. And then the strategy, if you're maximizing, so if you're always choosing what's the most rewarded, or the most frequent pattern in your environment, it almost becomes habitual, so it comes back in the putamen. So what we see in our MCI patients is that they in fact recruit this circuit that maximizes recruit. Now, when we've done these studies, we didn't do them in this nice chronological order. In fact, we ran our MCI studies before we ever developed the protocol to the point that we can do the computational analysis and figure out their strategies. So what we really need to do is go back and redo them and actually compute their strategies so that we know actually what are the strategies that MCI patients are recruiting.
Yeah, just a quick, maybe naive question. So the kind of learning you're talking about in the non-matching, the maximizing, is that a statistical learning like implicit learning? It would be the same implicit learning circuit that one might use in language and social?
Yes, indeed,
okay.
indeed. So what we have here is a case where, as I said, the participants are performing the task without any explicit reward, without an explicit learning. So it is very much like a statistical learning, like the classic statistical learning studies of language in very young infants. And interesting, what we see is that this learning recruits reinforcement learning circuits, reward-based circuits, which is a surprise in a way, but clearly what it shows is that there is internal reward in all of these tasks, even if you don't have an external teacher to tell you what's right and wrong. But it also reinforces, I think, the point that the brain really has circuits that perform computations. We come in and try to curve them up in different ways that make sense to us, but potentially the brain runs similar computations through these centers that involve reward or not reward.
I have a question on your first part on the MRS measurements for GABA. It's unclear to me whether when you did this, you saw that you had these two perception paradigms. So studies, for example, in rodents suggest that GABA levels change during learning as compared to consolidation, for example. So do you have dynamic data on each of your performances, whether what happens at the time of learning and at the time of that the individuals have learned the task?
Yeah, this is very interesting indeed. We are moving towards dynamic recordings of GABA. So in the human, at this point on a 3 Tesla scanner, to be able to extract reliable measurements of GABA, you need between 12 and 15 minutes, depending on your sequence. That's recording time, not streaming time. So effectively and this is voxel-based GABA. So although we are moving now to kind of more whole brain CSI GABA measurements. Now, we did these studies on the 7 Tesla. We started at Oxford, and now we're doing this in Cambridge. And we can collect from the 7 Tesla, because we have stronger signals, we can collect every 6 minutes. So in fact, we did these studies that I showed you, where you saw the curve going down or upwards. These studies were done every 6 minutes. We recorded. We recorded 2 voxels in alternation. So we be able to see whether it's local in one voxel or in another. But this is about an hour's session of training. So what actually we are capturing is the initial fast learning phase rather than the consolidation. It would be really great to be able to do this. There are considerations that need to be taken into account in terms of there is variability from day to day at the initial GABA level, at your starting GABA levels. So if you're looking across days, that's something to be taken into account. But we could potentially also take our participants out, train them, and put them back in the scanner and do the consolidation. So very, very interesting work. We are only at the beginning and very much interested in developing this further.
There's no other questions. Thank you again.
Great, thank you very much.